next section on describing color is systems, or our systems based on color matching. Now we're getting into more technical systems, more specific systems, and systems that are used more in the technical end of color matching. So how we actually go about working in industry, whether it's paint or plastics or textiles, inks, and actually working with color to match colors, get the right color that we want. So specifying colors using the previous systems can cause difficulty. The sample from a color order system exemplification appearance can change with viewing and illuminating conditions, so it may not always appear the same. Colors based on perceptual attributes without physical samples may not have enough precision and accuracy to meet specifications. May not be enough precision involved in it that we can't really use it for describing the colors that we want to make. One way to simplify color specification is reduce the problem to one of color matching. So we're going to look at colors in terms of a color matching type model, I guess you'd say. The color to be reproduced must match the color of the sample viewed and illuminated under a specific set of conditions. So we're going to say this is our standard. What do we have to do to get the same color? How do we make it? Where does the components come from? The term that we use for the instruments and that sort of thing is a synthesis of two words, color and metrium, Greek for to measure. It is science of color measurement, and we call it colorimetry. We can do it visually, or we can do it with instruments. We actually are measuring colors when we use our eyes. You can think of our eyes as color detection devices, and they're actually pretty good color detection devices. There are specific ways that we do that. It goes back to the 1800s and has to do with specifying the color of beer. So that may be of interest to some of you in this class, that you get the right color beer. That's important to manufacturers, and not only of beer, but if you go to any food manufacturer, whether it's natural foods or synthetic food, processed foods, they have color standards for their products. And if it doesn't match that color standard, it's rejected. They make cheese puffs out of extruders, pretty similar to the extruders we use in our lab, and it comes out just like any other plastic process, and they measure the color at the end of it to make sure it's right. If not, they adjust some of the ingredients. See, this is based on the principle of metamerism. If the same materials are not used in producing the visual output of colorimetry, the greater the chance of metamerism. So again, we have to use the same sources or the same colorants to get the same color all the time. Now, if we're only trying to match under one light source, then that's not as important. But if we want it to match under all conditions, then we have to be careful what materials we use to make it. We have a concept called the standard observer. Using an average observer would reduce the problem of metamerism caused by using a particular observer. Since everybody is a little bit different, we want to pick a standard person. I suppose you could go out in the population and test people and say, "You're it. You're the standard guy. We're gonna base everything on you." Standardizing the average observer would result in all specifications being constant, so that'd be good. So everything, when we talk about industry and we talk about quality control and that sort of thing, we want a standard that is good that we can compare our newest production to. Falls within the specifications that we set, then it's a good product and we can sell it. If it falls outside that specification, then it's not good enough. To begin to do that, we need to standardize on what it is our standard. What is it we're going to compare it to? So the CIE standard was developed, and it's kind of the heart of all modern color measurement systems. The CIE is a French acronym for an international color body that came up with this system. We reduce the problem to, does the sample match the standard? Yes or no? So in order to define the standard observer, which is the basis of all colorimetric measurement, experiments were carried out on human observers free of any color defect. They took a group of people First, they went through that Munsell test and came up with, a good result that they had no color defective issues. They weren't red defective or green defective or any of that. They fell within that normal range. That doesn't mean they were great at discerning color. They were just non-deficient. And they did an experiment. And this experiment is outlined in your book. So the observers were asked to match the colors of a test lamp using 3 primary light sources. So they basically had one light or lamp that they were looking at, and then they had 3 other lamps that they could mix together at differing amounts to get it to match. So this was all based on colored lights, again. Not pigments, not other things, but colored lights, so it was an additive mixing experiment. They decided on using these lights. These are the wavelengths of the lights that they were used. 435, 546, and 700 were the lights that they chose to use, and I'm sure in the book it tells why they picked those, but I'm not sure, and not that important, but those are the lights that they used, the red, green, and blue one. We had a reference light that was shined on one side of a screen. It was separated, and then a test field that we shined the other lights on and adjusted these till it came out to be the right color. And then they would measure the amounts of each of those lights or each of those primaries to decide how that color was made. So the test color was defined by how many of those primary lights or what amount of those primary lights were present in that particular color. Each observer was able to adjust the energy level of each of the primary light beams, the red, the green, and the blue. And then they kept adjusting them till both sides of the screen matched. And they would say, yep, both of those are the same color. Kind of what it looked like. And you can see right at this point they're not the same. So they would keep adjusting this test field here until it matched the reference field. And then they would write down what the individual ingredients were, or the power levels of each of the lights. From that, we developed what's called the standard observer. Now, the standard observer is not a person. It's not a person, but it's actually a set of numbers. It's a table of numbers. It's the curves that we use. Again, it goes back to mathematics, a way of modeling how we see color with using numbers. And when they first did this experiment, they used what's called a 2-degree angle of viewing. What that has to do with really is the size of the opening in the hole they looked through to see the colors. So that was actually a very small sample that they were looking at. I think they defined it at 18 inches. It was about the size of a dime. So they're looking at a fairly small color. And again, the 3 light primaries were those right there. Quantity of energy set for each of the 3 primary lights was recorded for each adjustment of the test light and at each wavelength. Test colors that they looked at were basically all the colors along the spectrum. So this enabled the response of the 3 color functions of the human observer to be established for the whole visible spectrum. So there would be an R number, a G number, and a B number for every single wavelength of color. And that's what the observer, CIE standard observer function looked like. On the bottom, we have the different wavelengths of light. And for instance, if we would pick, say, 480, we would see that it would take that much blue, it would take that much green, and it would take that much of negative red. So you all understand what the negative red is. We'll explain that in a bit. But there is, we actually call it negative light there. So based on the actual trichromatic functions defined by the previous slide, mathematical transformations were developed. Led to the identification of a color stimulus by means of 3 numbers. Mathematicians don't like negative numbers. They said, we're going to get rid of that. And they came up with these curves to represent it. Now going back to what the negative number means is that they found out when they were doing the tests that there were some test colors that they couldn't reproduce with the 3 lights. They found out they just couldn't get it. So what they fooled around with and what they did is they found out if they took one of those test lights and moved it over to the other side, the standard side or the test side, and shined it on that sample, then it would match. And that's where the negative light came from. They're actually taking it away from the original test specimen. Kind of a strange concept, but it's not the most important part of it. But the point is, we now have 3 mathematical functions that can be used to describe any color along our visible spectrum. Now, since we did a mathematical transformation, they're not R, G, and B anymore. They're X, Y, and Z. And you can just think of them basically as numbers that sort of kind of basically match how our 3 different cones work, our red, green, and blue cones. They're a little bit changed mathematically. Doesn't change how they work. It just makes it easier to manipulate them and easier to use them when we do our calculations for other things. The previous slide was adapted in 1931 as the CIE Standard Observer Function. And they eliminated the negative values that made it hard to do the math, so they just changed it so that it could be used easier. They do not represent real functions, but rather average functions which are representative of a human, average human observer, a standard human observer. It's based on a 2-degree angle of vision. We'll see that there is another angle of vision that is actually more accurate that we will use when we get into the lab. And that's the 10-degree observer, which this one is, which was adopted in 1964. Back then it was later proved experimentally that the result of the colorimetric calculations at 2-degree angle of vision were not in perfect agreement with visual observation. So they did some other tests, did it with this 10-degree observer, and the data actually matched better with visual perception. The idea there, or the theory, is that when we look at samples, we don't look at little dime-sized samples, we look at bigger samples. Part of the light falls on our fovea, but also part of it falls on just our retina where there aren't color sensors. That combination gives us a slightly different perception of the color that we see. So when we model it using the 10-degree observer, it gives us better data than if we model it with 2-degree observer. So again, we're trying to model what's actually happening in our eyes and our brains and all that with numbers. So we're trying to match that. So this 10-degree observer matched better than the 2-degree observer did. Then we can, just to give you an example of what the difference between the 10-degree observer is and the 2-degree observer, this is relatively the difference between them. So if we think of that middle circle as a dime size, you can get an idea of what the 10-degree observer size sample is. If you look in your book, another good reason to have your book, you can see the actual size, see it up on the screen. The next slide shows the difference between the standard observer functions for the 2-degree observer and the 10-degree observer. There are some differences, not much difference between them. We generally use the 10-degree observer in industry, and when we do our measurements in the lab, we use the 10-degree observer. Now, is there a huge difference between them? Does it really affect when you're matching colors? Sometimes yes, sometimes no. It all depends on how critical your match is, what colors you're dealing with, what the material is, all those sort of things. But the general accepted norm is that we use the 10-degree observer. Whenever you're reporting numbers for this sort of thing, you always need to specify which observer you use, the 10-degree or the 2-degree. And we'll see that later on as we go through. From that, we can calculate what's called tri-stimulus values. And those are mathematical functions of the standard observer that allow a spectrograph to be converted into 3 numbers known as X, Y, Z tri-stimulus values. So what we're going to do is take that observer curve that we used in our calculations that we did earlier today and replace it with our standard observer curve. So we'll take that one curve out and put 3 curves in. Then do our calculations. And when we do that calculations, we come up with 3 numbers, x, y, and z. And capital X, Y, and z is important because there's also small x, y, and z that are used to describe a different set of numbers. But they're called the tri-stimulus values. Every color that we can identify has its own set of x, y, and z numbers, its own set of tri-stimulus numbers. So they are calculated in the same way that we did in our exercise, but we replaced it again with those 3 functions instead of 2. And the reason I didn't have you do this is it's a lot more calculating. Start off with, at the top of the page there, with our light source, like daylight. And then we have our object curve on the right top, which looks like a phthalo blue color. And if we mix those together, multiply those together mathematically, we get this curve right here in the center. That is the light source multiplied by the object. Then we take that curve and multiply it times the x curve, the y curve, and the z curve for the standard observer. Remember, it's not the RGB anymore, but x, y, and z. And then if we come to the results that are the last set of graphs here, we have capital X, capital Y, and capital Z, which are tri-stimulus values. And the actual numbers are calculated by the area under those curves. And I'm sure you all are very familiar with the techniques for calculating the areas under the curves. Correct? You've all had calculus. That's pretty simple stuff. If you haven't had calculus, I guess I won't have to have you do that. Well, just know that's where they come from. But anyway, if you ever wanted to know how you would use calculus for anything, this is one example of how you would do that. There's lots of other ones. Calculating the area under the curve can be done using calculus. By going through this process, we can identify any color that's made up of a certain light source, a certain object, using the standard observer, and give it 3 numbers to define it. Our tri-stimulus values, capital X, capital Y, and capital Z. If we put it in terms of the graphs like we showed earlier, we look at it this way. We now still have our light source curve. We multiply it times our object curve. We multiply it times our 3 standard observer curves, and we end up with an X, Y, and Z value, tri-stimulus values. In this case, they're blues, like I mentioned. Our X is about 13, our Y is about 14, and our Z is about 47. And if you didn't see that was blue, could you tell that by looking at those X, Y, and Z values? If I said X was 13, Y was 14, Z was 47, you would say, of course, that's blue. I can't do that. And nobody can do that. They are numbers that represent a color, but they don't mean a whole lot to us intuitively. We'll see later on we'll have numbers. If we look at them, we'll actually be able to get an idea of what the colors we're talking about by just the numbers, especially by difference numbers. But we can't do that with X, Y, and Z values. There's no pattern to them. There's no real rhyme or reason to them. But technically, that's the start of where all our calculations come from. That while these numbers identify colors mathematically, unless you have an incredible memory, they really are not in a form that's very useful. You have to convert them into other numbers to make them meaningful.